So to remind you, we've been talking last week about kind of doing two things at once. Asking a whole set of questions of what we might want to know about face perception in the brain. There's some questions. But at the same time, the agenda has been to consider the different methods available in human cognitive neuroscience and what kinds of questions each one can answer. Okay? And its ability to tell you whether the neural activity you're looking at is causally involved in behavior is like nil. Okay? Now, at least one of you, I only read a few of the assignments, but at least one of you was fancy magnetic sensors in the big hairdryer device around your head. There you get beautiful temporal resolution, but it's like, you know, the Heisenberg principle of cognitive neuroscience. You want time, you don't get space, right? Okay. And similarly, here we can measure the causal effect of a patient who can do B but not A, say recognize faces but not objects. And when you have in the literature two cases like that, now you're in a really strong position to infer that there's something fundamentally different about face recognition and object recognition in the brain. Okay, so that's really important. And you guys know from 802, another case of the right-hand rule coming to our service. You have a hell of a current going in a coil. What's going to happen in brain tissue underneath? Yeah, exactly. And on the scalp, what is the closest spot to that region you found in their brain previously with functional MRI, and stick the coil right there and exactly titrate its location with reference to that brain image. Okay? So that makes this whole great, because in this case, we're testing the causal role of a bit of brain on behavior. So we're going to measure behavior. And so what is our task? Okay. So here's his task. Sorry, it's a little tiny, but this is one trial. Time is going this way. You present a face. There's a brief interval. You present another face. And the task is just, are those two faces same or different? Okay, it's kind of your basic. Noise, and it's kind of like somebody went like that. So you might imagine you need a control condition, right? Because if people are that also has a TMS pulse, right? If you bang somebody on the head when they're trying to do a task, you probably disrupt their performance. We talk about activations in the brain, like those little yellow patches I showed in monkey brains a moment ago. That shows you the bits that responded more in functional MRI when that monkey was looking at faces than objects. The contrast is faces versus objects, right? It's looking for a higher response in one condition than another. Remember, importantly, I said this briefly way back, functional MRI can only tell you about differences between two conditions. Right? The absolute number, you're going to measure the MR signal intensity in one condition, say when people are looking at faces, and it's going to be something like 726. And it's totally meaningless a little bit, but this is the crux of the matter. What are those conditions, and are they the right kind of minimal pair? Okay? And you guys got the gist, but I felt like most of you didn't really engage. Okay, what exactly were those non-snake conditions, right? So that's really the crux. So all of that said, minimal pairs are kind of like a platonic ideal of experimental design. They're what you aspire toward, but you can never really do it, right? If the two conditions were identical, except for this one little thing, they'd be identical, right? You can never totally pull it off, okay? But you can track the little ways in which you fail. What we're going to do. We're going to break into groups, and you guys are going to think how to take the kind of designs that you already put together and turn them into actual experiments, which is going to require deciding on a whole bunch of other things, and then we're going to discuss the things you come up with. Okay. Or is it a waste of scan time? Think about that. Okay? Okay, next, suppose you get to scan 10 subjects for 1 hour each. Now think about how that design is actually going to go. Are you going to assign different conditions to different subjects? So these for the moment. Okay, so we're going to break you guys into 4 groups, and you're going to talk amongst yourselves and try to come up with your best answers to these in like 5-10 minutes, something like that. And then we're going to pool your thoughts on this when we get back. Okay?
Why? Oh, because you know, when you see a snake, it kind of slithers and has this very distinctive thing that it feels like the motion is what creeps me out when I see a snake.
Totally.
And,
Me too. Right.
So one idea was to have them, like essentially indicate if they saw a snake or didn't see a snake, but that brought up problems like, well, we don't want them thinking about snakes the entire experiment. So
Also, if they're going to tell you what a baseline condition was, sorry about that. What I meant by a baseline is different from a control condition. A control condition would be like the non-snakes contrasted with the snakes, right? Baseline tends to be like a minimalist condition that's supposed to, turn the brain off. Okay, can we turn the brain off?